My name is Corinne Teeter. I am currently at the Allen Institute for Brain Sciences. So today we have a slight change in the printed agenda. I'm going to give a talk first for about a half an hour, and then after that, Philip Meyer from Brain Corporation is going to speak. After that, Jim Olds has agreed to come up and talk about maybe future steps after this conference. We'll take a short break, and then we have John George and Jennifer Hassler, and then we might have a surprise guest from Sandia come and give a talk on their MemRister technology here, but we're not sure whether that's going to happen. If not, we might just open up the floor for any final comments and then adjourn the meeting. So I will go ahead and get started. So my background is in computational neuroscience, so I'm interested in figuring out how the brain works, and very specifically in figuring out the basic principles that allow networks of neurons to perform computations. And I think that the technology aspect is very interesting. Once you figure out these basic principles that are in the brain, then hopefully you could apply them in places where they might be useful in technology. So what I'm going to talk about today, though, is something, it's a very simple proof of principle, and unlike the people here, it's not going to be dealing with large networks of neurons, although at the Allen Institute we are clearly going to model large networks of neurons, the visual system of the mouse. But before I get started, this work kind of touches on 2 points that I think might be important when thinking about making brain-inspired computing. And one of those things is, and I'm glad that it got mentioned yesterday by Miko specifically, the level of abstraction of which we have to model this system. I mean, as you saw in many of the talks, the brain is an extraordinarily complicated circuit that is generally modeled by point neurons. And it's unclear whether or not that's the appropriate level of abstraction. And so I was very excited to hear Hans Meyer say that he was going to create very complicated networks of neurons and then reduce them to see when the function starts to break down. I think that's very important when thinking about this stuff. The other thing that I just kind of wanted to mention is that the brain has a lot of variability, and there's growing evidence that says that this variability might actually be helpful for doing computation. So that might kind of take some of the constraints off, making components that are actually exactly the same or throwing them out. Maybe actually variation in components would be a good idea when going forward and trying to make these systems. So what I'm going to talk about today are can ion channels overcome the biophysical constraints of morphology? And I'll show you a little bit what I'm talking about. So I think what we're all trying to understand is what this messy circuit does. Now this is a very ugly picture. It's a Golgi stain of human cortex. Many organizations have much better visualizations of this. But I think it kind of shows what people have been working with over a long time span. So this Golgi stain, which means it only stains a very small percentage of the neurons in this circuit. And as you can see from this, it's a mess, and so we're trying to figure it out. And so in order to figure out any circuit, you would want to identify what the components are and how these components fit together. And hopefully, since the brain is so complicated, you would want to understand what the underlying organizational principles are. So the brain is a particularly interesting circuit in that the structure of the components will influence both the function of the components themselves and the connections within the circuit. So Jim talked about how this creates this fascinating structure-function relationship that we don't quite fully understand. So neurons come in very different shapes and size. This is just an example, and these are only the dendrites of a few different types of neurons. So you can see there's lots of variation there. And if we know anything about neurons, we know that the membrane itself, they can be looked at in and of themselves as little circuits, right? Because their membranes have resistive and capacitive properties, which means if you inject a current, say at a synapse, into one neuron, it's like injecting a current into a completely different circuit than if you inject it into a different neuron, right? It has different circuit properties, different input-output functions. So this was kind of elegantly shown in this paper by Meynens and Sinofsky in Nature in 1996, where they took a bunch of different morphologies, held the ion channels constant, and showed that you get very different spiking behavior from the different morphologies. And if you sit and think for a minute about what that means for the implications of a circuit where you have, all of these different types of neurons are trying to communicate with each other, you realize that if they're all communicating with different input-output transfer functions, they're going to be different populations of neurons talking to each other in very different ways. So, of course, this isn't the whole story. Neurons also have ion channels, and it's ion channels actually that make the spike happen. And one of the interesting things about these ion channels is that they do vary between neurons, and actually neurons can vary them to achieve the spiking behavior they desire. Like this has been shown. If you take a neuron in culture, you change its spiking behavior, it will adjust its ion channels to achieve its desired spiking behavior. So there's homeostatic mechanisms at play. So what I wanted to do is I just wanted to know whether ion channels could overcome the constraints morphology places on the spiking behavior of neurons. And if they can't, it'll probably be pretty important to take into account the effects of morphology in these complicated networks that we're creating that are usually point neurons. If they can, it's I would never say that the brain doesn't use morphology to do computation. But it suggests that the brain has quite a bit of flexibility with regard to its shape and information processing abilities. So the way I went about doing this is I just did a very simple thing. I wanted to see if I could find a set of maximal conductances that would allow one of these neurons, say, this small layer 3 a spiny cell, to fire like this layer 5 pyramidal cell. So I wanted to take each combination to see if this neuron could fire like this neuron, if this neuron could fire like this neuron, if this neuron could fire like this neuron, to see if the brain was actually able to do that, to achieve that. So the way I'm going to do this is I used a genetic algorithm, and I allowed the maximal conductances of the ion channels from the original paper. So there's 5 ion channels: sodium, potassium, slow and activating potassium, high threshold calcium, and calcium. And so I just took those and I let them vary separately over the dendrite and soma. And it's kind of interesting to note that I wasn't able to get the results if I didn't let them vary separately over the dendrite and the soma. So maybe that says maybe you need at least 2 compartments. I don't know. But so I let those vary by a factor of 10, put them in a genetic algorithm, and then what I did was I had a fitness function, which is a target voltage trace. So this is the voltage trace from that layer 5 big pyramidal cell, and simulated the behavior of these neurons using a neuron simulator, right? So it takes into account all the biophysically realistic ion channels, right? Neurons simulator takes the maximal conductances given by the genetic algorithm, sends back an obtained trace, and then those traces are lined up, and a fitness is calculated basically by taking the peaks of the spikes. So basically only spike time is important here, not the subthreshold currents. And right, this whole thing iterates just like a genetic algorithm. And so here are the results. What I'm showing you are the original traces from Meynens and Sinofsky in blue. And what I'm going to show you in red is the best thing I obtained using a genetic algorithm. And so my best fits are overlaying the original. And as you can see, basically in all cases, a neuron was able to find a set of maximal conductances that would allow it to fire like this with this fast tonic spiking behavior. Again, we see the same thing for the layer 4 cell. All of the neurons were able to achieve the spiking behavior of the cell. Again, for the layer 3 pyramidal cell. Where there did start to be some problems was in this layer 5 pyramidal cell. So what I'm showing you here is the insets of the bursting activity. And basically in all cases, the smaller neurons were unable to slow down their frequency of firing to achieve the firing behavior in this neuron. And that may not be surprising because there is theory about how these neurons actually produce bursts. You have the spike is initiated in the soma, it kind of travels up the dendrite, reflects off this. Meanwhile, these sodium channels are resetting themselves and then comes back down and fires another spike. So in this case, the morphology might be mediating this behavior. And of course, in real neurons, they have much more degrees of freedom than just altering the maximal conductances over the soma and over the dendrite. So maybe in a real neuron, it could actually do this. Unsure. So just wanted to say a little bit about what the ion channel was that was actually allowing this to happen. So most of bursting behavior is thought to be controlled by high threshold calcium. And so this is a nice figure here, and all of the neurons, basically what I'm just showing is I'm adjusting the amount of calcium, right, from 0 all the way up to some number for each neuron. And you can see that as you alter the calcium, you take any neuron from a tonic firing regime to a bursting regime. Here a little less so, you kind of have doublets even when there's no calcium. So that kind of shows you that this neuron really does want to be in a bursty regime, but still, you know, you start adding calcium and you go from this kind of spiking behavior to more bursty behavior. So in summary, basically I just showed that the spiking behavior of the smaller neurons can always be fit by the larger morphologies, and the spike bursting or the spike intervals of the larger neurons could not be fit for the larger neurons. So of course it's possible in a system where there's more ion channels that this could overcome this. I don't know, but at least that this is some hopefully positive evidence that maybe modeling a system as a set of point neurons is not completely wrong, which I don't know, I find to be a good thing. And that's all I wanted to say today. Thanks. Is there any questions? John?
So it seems to me that, that in that large neuron, you know, the one clear difference is that it's a lot larger, but the other difference is that it's really kind of bipolar in its functionality. Mm-hmm. Is there any evidence that that's the thing that really contributes to that behavior?
Yeah, I mean, yeah. So there, you know, there are theories that show that there's actually, I think, a calcium hotspot at the top of the apical dendrite that's causing dendritic spikes. But basically that mechanism that the spike, you know, travels from the soma up to the dendrites, does something there, and then travels back down and bursts again is, so at the Allen Institute, for example, when we're doing our large network models, we're starting out with simple neuron models, single neurons, but like those neurons in particularly, we're going to put in morphologically realistic neurons because we think that their function is really more complicated than something that simple. So, yeah. All right. Anything else? All right, so next Philip Meyer is going to come up and speak about his research.
And today I'm going to be talking about inhibition in a spiking model. Particularly, it's a model of motion processing in cortex. And my background actually comes as a physiologist, so I've done experimental work in rats, behavioral work in animals, looking at inferring how we can infer function about networks and systems, the visual system particularly. And so this is a shift for me into an entirely in silico simulation of a system I'm familiar with, but now I have a lot more depth into being able to measure every single neuron in the system, designing the neurons to have the properties that we think are present in the biophysical models that we're looking at most of the time. But of course, when we're approaching it differently, we're loading in everything that we sort of want to have into the network and confirming that it has the properties that we think it should have. So it's a different process in the sort of reverse engineering to have what you think it should have, confirming that in fact it has the properties that arise to it behaviorally that you think it should have from a low-level dynamical system. And one of the challenges that are present, because we've taken this stance, do everything with spikes. So all of the nodes are doing spiking computation. You come with all the challenges that spikes have. So neurons will saturate at a particular level where they can't fire faster than that. So you can't send, if you wanted to do rate coding, you'd hit a maximum amount. They'll also have a floor. Well, they won't send anything. If you're just tickling them, they'll sometimes emit no spikes, and you're getting no information through there. So this could be a benefit from a communications and an energy point of view, but it ends up determining how much information passes through your network and how much you're computing with. Means that you want your neurons to sort of be in that sweet spot where they have some type of graded capacity to communicate information, whether that has to do with a shift in latency, an increase in the total number of spikes. You want to make sure your neurons are in that range. And one of the challenges there that's going to fight that is that the world contains complexity. Sometimes you have very strong driving signals, either a high contrast thing or in the case of designing a cell which is sensitive to certain motions, for example. You might be tuned to only certain properties and not to others. Occasionally there will be strong driving things, and often there will be weak driving or non-driving things. And you still want to communicate information across a range of these characteristics. So to do that, we know that the brain has inhibition, and we know it's sort of capable of setting that point dynamically to be able to still encode, despite the variability in the context of what's going on. And so my focus here is on trying to get sort of basic motion processing through the system and how it is that you end up being able to benefit from inhibition to get the job done. So just a brief moment on Brain Corporation. We are a company that has two sort of core focuses, vision research, perceptual systems, stuff that's close to the periphery, and then close to the periphery on the other end for motor control. I'm on the vision side, and that's all I'm focusing on. And we also work on technologies that we want to enable. And this is fundamentally a research talk and a research focus. So it's sort of basic science, what we can enable by virtue of spiking neurons in a network. It's work that I've done with Micah Richard, Jairam Nageswaran, and Eugene Iskevich. And this is all motion processing work at Brain Corporation. So for the dynamics of our individual cells, this is one neuron in a large simulation, but what I've pulled out here over 4 seconds is just sort of a trace of the voltage that's fluctuating. And as you can see, often it's just fluctuating at a voltage that's very subthreshold. Subthreshold fluctuations. Occasionally there'll be a little bit of a peak where it might be ramping up to get close to threshold. Threshold's actually above this. And here, throughout this 4-second period of time, there were 4 spikes that occurred, one here, here, and here. If you're familiar with some of the dynamical systems models that Eugene Iskevich has worked with, these Iskevich neurons, they're two-dimensional models. And to give you a point of reference, a leaky integrate and fire neuron would be a one-dimensional model often, where you're just modulating something like voltage. A Hodgkin-Huxley simulation might have sort of 4 dynamical parameters that are updating and influencing one another. So this is in the class of a two-dimensional model, has a little bit more complexity than what you'll find in leaky integrate and fire. It's not doing full biological realism. But it's doing something that phenomenologically captures a lot of the states of neurons. We can get more of the dynamics after resets and more of the dynamics for adaptation. And so this is just the two-dimensional state space. I've just plotted this exact same trace, but as a scatter plot over time. And what you're seeing here is this is when you're at rest roughly as little bits of activity are bombarding it. It's sort of either oscillating around down here or bouncing up and down. Occasionally a spike will be fired, which will be a trajectory, which will pass over across a threshold. And afterwards we'll have a reset, which actually ends up somewhere off this chart, which will then end up adapting back over to here. And so this is sort of this adaptation component. The slow time scales are encoded in U, and these are these typical voltage axis where it's spikes coming with high peaks in that direction. So these are the dynamics in every individual cell. And you might kind of want to say, okay, well, if I've got one of these neurons and it's being actually driven by a natural stimulus, there's some input we're loading in, a digital image, what's actually going to make it spike at a given moment in time? What are the main properties that are going to influence it? So we have these two-dimensional state. There's 4 parameters that end up determining the dynamics of the cell. We're mostly choosing a small subset of the total space of parameters. So a lot of our neurons actually have the same parameter choices. And so it's the drive that's coming in, the current, which we're doing from a conductance-based model. And at any moment in time, if I want to take a guess, well, is it going to spike now? Is it not going to spike? Really, I'm reasoning about the total amount of excitation that's arriving there and the total amount of inhibition. There's some nonlinearities that are going on, but still my reasoning is how much is going on recently in time, how much excitation, how much inhibition. And so over time, this is a short sequence of time. This is the amount of, what I'm showing here is now conductance, which is coming from 2 sources, either the AMPA, the excitatory conductance that's coming in from the drive to the cell, as well as GABA, and this is coming from feed-forward inhibitory, as well as lateral inhibition in the network, is producing sort of temporal traces that look like this. You'll have these volleys, fluctuating amounts of conductance, and the inhibition here, you'll have these sequences of inhibition that are roughly matching in their temporal time course. So this is a principle that we observe and we see in many cortical areas. It's present in cortex, in hippocampus, in colliculate. In many areas, we've observed these phenomena where the amount of excitation rushing in and the amount of inhibition are balancing one another out. And it seems like it plays a very important computational role. There are many possible ways you could exploit this to get some work done. But in this case, I just sort of want to reason through. So this is a real trace that I have from somewhere in the middle of my simulation. I added in these, I didn't record the spikes from this particular one, so these are fake spikes that I've added in here, just explanatorily. You can imagine from a drive like this, you might produce 2 spikes. There's a lot of drive, no inhibitions holding you back. In this case, maybe this excitation is enough to be able to push you to produce 1 spike, but then this really isn't enough to be able to overcome the amount of inhibition that's going on, so you'll only produce 1 spike and early on. And in this case, it might be that so much inhibition came out that even though you're still getting some drive, just the cell's silent, nothing's happening. And in a case like this, you might get that early spike right here based on your initial amount of excitation. And then at a subsequent moment in time, the network sort of is capable of balancing. You could almost think of this as a prediction for how much the total magnitude it's trying to cancel out. But yet this is higher. This is sort of uncanceled excitation. And it's the difference between these 2 things, the fact that there's more excitation here that may allow this to provide a sort of a second spike. And so this, you could think of these as, maybe that's not that informative. These sort of are happening every once in a while. The fact that this happened right here, that's pretty meaningful. It carries, not only does it bear information about this driving event, it also bears information about the fact that this was larger than some internal event. This allows you to compute a lot more with a spike. So why do this balancing? I'm just going to suggest a couple ideas right here. We're not exploiting all of these, but it's kind of relevant to think about it. If you sort of cancel out everything, you can drive the network into a critical regime where sort of everything matters. Every little bit of inhibition that was capable of canceling it out is determining whether you're above or below that threshold. So you're putting it at a point where you really don't actually know whether it's going to spike or not, and potentially that spike can bear information about a range of different things from a range of different histories, different contexts that it's indicating. You might be able to exploit this to actually get a stochastic-like nature to a network. That might be useful in some cases. You could also exploit something like this to be able to manage your gain in such a way that very strong excitatory drives, which would normally be saturating your neuron to sort of a maximal firing rate, are not going to be because you've got the sufficient amount of inhibition that you still have some gain, some leftover room to encode what's going on in your stimulus. And what I alluded to, the notion that you could think of inhibition as a prediction of what's going to come, the amount of excitation that is going to come. As a result of that, you could actually think of the difference between the two as maybe encoding something like an error signal, what was unexpected. It's not exactly like an error signal fully, because if you were below it, you're not firing, and you don't necessarily get the magnitude of your errors in the reverse direction, but there is something like an error signal that you could build into a network like this. And then redundancy reduction. If you imagine you have two stimuli that are potentially encoding similar things in two neurons, and if one of them gets there, the network kind of already knows it, and it's able to sort of send some inhibition to a neighbor, which was going to deliver the same message, and say, hey, the network already knows this, so you be quiet. Don't resend that information this time. And so you could find a way to sort of cancel out the spikes that would be carrying the same amount of information, and therefore the entire volley from a population might be more information bearing. So there's a way you could use inhibition to do that. So in this case, I'm definitely not doing all of these different things. These are just sort of ideas I wanted to put forth. I don't feel like these were, these are almost, you could think of them as if you were designing a neural code, and you do, so biology probably has one, different areas have possibly particular ones. We don't know what they are, but if you have sort of the luxury of saying, I'm designing a network, I'm going to use certain principles, you might be able to exploit these different properties to get the desired state that you want. Okay, so a little bit more from a network point of view. This is the sort of the level of abstraction that gets at the 4 different brain areas that we're modeling, from the retina, the LGN, V1, particularly we're emphasizing 2 layers of V1 in the motion system, 4C alpha and 4 beta, which are 2 bands in here. There's the traditional 4 to 2, 3, and 5, 6 network is not used as much for the motion processing system. It's sort of got a two synaptic hops in V1, and then it pops right up over onto MT. And so we can look for inspiration in this system and say, okay, what level of abstraction are we going to accept here? And so this is about the level of complexity that we're getting at in terms of the number of types of neurons within the different areas. So in this case, I just want to bring your attention to these 2 areas right here, which I mentioned. This is one layer within V1 and another layer within V1. There's an excitatory population, which has basically got the main excitatory drive, which is passing through here, and 2 different types of inhibitory populations, which are then dumping inhibition. And it's this synapse right here and this one right here, which is generating what I showed you. That was the balanced inhibition, which is sort of matching out the drive which is coming into the network. So one other attribute here, right? Like, so I'm showing you one node right here in a real network that how many neurons is that? What is it actually encoding? So we've designed this to have 24 channels, each channel spanning tiling the full space of the retinal visual field that we're modeling. So it's a 6 by 6 degree visual field. And across that, these sort of hand-designed channels will span speeds and orientations. So 8 orientations we've sort of hand-dialed into there. And that's entirely based on the properties of the layer 4 synaptic weights. They're doing something like motion energy encoding. I'm not going to go into that in too much depth. You can ask me afterwards. But basically, it's a phase-shifted Gabor is being able to pick up the translation of something for a particular spatial frequency. So depending on the speed and the scale of a moving thing, you're capable of generally picking up the motion energy. And this is consistent with the biological models for how we think we're getting motion information into the visual system. And that's sort of hand-dialed into these weights right here. But then the sensitivity to that is not great. You'll saturate a lot and you won't encode very well. And so what the inhibitory network is doing is it's sharpening and it's increasing the dynamic range to be able to actually, for a range of different circumstances, be able to appropriately integrate this information into what is something like optic flow in MT, where there's the translation of moving information, and you collect information from a spatial region, you integrate it up, and you still want to know with some fidelity it's in fact moving in this direction at this speed. So the end answer is speed at a particular direction. That's what you'd like to be able to get out of a network like this. So to make this a little bit more intuitive, I'm going to show you one of these channels in layer 4B. So that's basically the excitatory guy right here. We made a little movie. And normally, we're actually using pretty simple objects for decoding. It's a rigid object, which is translating linearly. But I'm going to show you a natural movie because that's a little bit more fun and lets us sort of think about it. So in this case, you can see the activity that we're seeing here. Each little small point in green over here is the activity from a single spike on a particular frame. There's a relationship between frames of the video and the clock on our simulation. So we're doing 40 steps of our simulation for every frame Each one of those is a millisecond, and I'm just saying if any one of those frames contained a spike, let's draw a single point right here, and then so you can kind of visualize the total spatial activity in one of the channels. So things you might notice, at some moments in time, so there's like a scene transition right there where something kind of, it faded in across the scene. So there's actually a lot of activity spatially across a bunch of different areas. There'll be some spurious things here and there where it's picking up small motions in the trees, but generally you'll get a lot of spatially contiguous motion information from the moving skateboarders. I want to show you one more where I've slowed down the speed substantially so you can pick out the per millisecond updates, basically what I've hidden from you by virtue of showing you only a spike per frame. And so this is just a short clip of the same movie where you can make out the sequence of the arrival times of the individual spikes. So in this case, what you're seeing, that was one frame. Here's another frame. And there's a temporal process by which there's the early arrivers and the late arrivers. And so information is being conveyed. And here, the simplest one is something like contrast. The strongest drives for the sensitivity of these cells come early because they're capable of integrating faster up to the point of the threshold. And the weaker drives are still there, but they're a bit weaker. And so they'll arrive later in time. And that comes into the processing mechanisms for the dynamical system. OK, so I've described to you how it is sort of the spirit of what we're loading into layer 4, that it's following this motion energy model. But I didn't tell you anything about the inhibition and how we're figuring, there's a lot of different weights in here. Are we hand dialing all of these things as sort of this massive exercise in twiddling lots of knobs? And so what we're doing is we're letting a spike timing rule fit all the inhibitory weights to be able to get at that balanced threshold. And so the rule is fairly simple. The spirit is something that we want it to be local so that the information that's present in the postsynaptic and the presynaptic cell is basically able to say, well, sometimes this one's firing a lot, and when it fires more, it's OK for me to crank up the gain under those contexts, and when it's firing less, I shouldn't because I don't want to shut it off. The curve is in many ways sort of non-biological. You'll see sort of these exponential tails sometimes. There's this classic B and Poo result where you have these different shapes in here. There's some recent results suggesting that if you have these sort of symmetric Mexican hat ones with suppression on either side, you can get some inhibitory matching. But what we found from using a bunch of different learning rules is that often you can get quite far with just sort of like little boxcar things. The important thing here is that regardless of how long ago, so this is sort of like, this is time agnostic in this sense, which is every time this neuron has a firing of, if this neuron is firing too much, then it's OK to be able to turn it off more, and if it's not, you're more sensitive in the other direction. So you can read this as, in the short window where I was capable of being able to turn off the cell because the excitatory spike followed shortly afterwards, it's OK to bump up my inhibition. So this is going to increase the amount of inhibition in this synapse that this cell is delivering onto that cell under conditions where they fire proximal in time. But in other conditions, when there's just spiking going on, you'll get sort of this weak suppression that happens so that there's a downdrift in the other direction. The end result of this is these will sort of match out, so the frequency of occurrence of things that increase the weight of the synapse and things that decrease the weight of the synapse, in the stable state, they'll cancel one another out. So there'll be slightly fewer of these. This will happen less often, but it's stronger in magnitude, and they'll cancel one another out. OK, so I wanted to share one problem that happens problem you can run into when you're doing this. So you'd like to be able to run your simulation fast, and so you've chosen a particular time step for your simulation. But you also want a lot of context to be able to influence sort of that first spike or the first volley that's getting in there. So ideally, you'd like to allow lots of inhibitory integration to get in there to sort of do the right canceling. But you've only got one time step to be able to send all that spiking information to be able to integrate it and then cancel it out. So this is, maybe biology doesn't have to worry about this because it can operate at different time scales. Maybe this is just sort of an artifact of our simulations. But there was a biological solution, and I'm sort of sharing with you a digital hack of how we're addressing this problem. The biological solution might have been to say, look, sometimes this lateral processing, it takes too long to be able to get these guys to actually activate lateral inhibition. Lateral inhibition comes later. At this point in time, this population of excitatory neurons is already sort of done firing, and so you can't influence their code anymore. So you can use something like feed-forward inhibition, where there's a really fast volley, where the inhibition is actually capable of arriving. Again, you have to worry a little bit about this latency. You don't want it to come afterwards, so this latency has got to be really fast. and there are cases where it arrives like almost synchronously, that you can kind of get it there really quickly. We don't have that luxury in this case, and so what our solution is actually sort of going to be to stretch out the evolution of this response by forcing there to be jitter between the arrival times at a synaptic level. So what I mean by that is the arrival of these guys, basically everything with a red arrow over here, the excitatory drive onto this excitatory population, as well as this feed-forward drive onto this is sort of like a feed-forward inhibitions being delivered like this. These are going to be jittered in time with something random but consistent per cell. So this cell, which is part of the excitatory one, has sort of a fixed onset latency to it. And what that enables it to do is that when the system has some inhibition, it can reliably be able to predict if this guy was going to fire here at this moment in time. And importantly, there's already something going on in the network before this. So it's capable of establishing a context and then delivering some inhibition in order to be able to address that. So another way of saying this is when you get later into the response temporally, there's more capacity for the network to sort of cancel itself out and to compute. And we've stretched out the range at which it can do that computational by using synaptic delays in a particular way. So this might have some interesting sort of design principles from hardware. If we really wanted to implement this, there's some trade-offs for speed and how you implement it. And so just to bring this back to one of these guys, this is like saying, okay, yes, at the beginning, I might have some excitatory drive, which is sort of uncancelable. It's hard for me to cancel it out, and it conveys a certain kind of information, whereas later on, this is potentially contextually informed. There was the room to integrate and cancel it out, and so this might be a smarter response. The code might actually be a different code at the end of the response compared to the beginning of the response. You might sort of multiplex your code over time. But this is one of the things that's sort of potentially informative and information-bearing. And that's one of the things that we're making use of in this model. So I really only have 2 more slides right now of data. So I'm going to show you the tuning of an empty population and some decoding we've done from the network. So what is this here? This is a matrix which is encoding basically the firing rate of a population of cells for a range of different stimulus conditions. So we're sweeping through all different speeds, uniformly choosing speeds randomly, and all different directions. And then we're asking this population of cells, does it have a stronger evoked response for particular speeds and directions? And yes, of course it is. It's most tuned to 180 degrees, and it's most tuned to speeds around 5 degrees per second. But it's a little bit tuned in the off directions. There's a little bit of tuning in this direction, and a little bit of tuning in this direction, and that's good. That's what we're going to be able to exploit for the decoding. So in designing a network, we're looking a lot at this, and then in terms of decoding it afterwards, we're fitting it with something simple. In this case, we're just doing regression on it. So this is a diagram which is sort of trying to convey a qualitative sense of the amount of noise in the system from a single sample. So what I mean by a single sample is if we have 3 frames of contiguous activity, something translating 3 steps like this, that's the least amount of information that we can start getting a reliable motion signal out of MT. And so, of course, if we did this longer, we could integrate, we could have some prior over acceleration or speeds, we could clean this up a lot, but I'm showing you sort of one independent sample. How good can you get at decoding the direction from a range of different speeds and directions? So this would be vertical motion here, sort of at 12 o'clock, this dot right here, and so you could say, well, how good are we at determining that it was purely vertical when it was purely vertical? Well, here are sort of 5, these are sort of the individual samples that we're looking at, and this is showing you the average location. We do have a little bit of a bias. We're encoding 12 at like 12:30, so we're not perfect in our estimates, but this topology is being preserved in terms of our direction decoding, and we have a little bit of noise in there. And sort of my last point that I wanted to make here is that we're actually intentionally using a very easy to understand and not the best decoder. It's linear, and that's sort of encouraging from the point of view that a single neuron could have a bunch of synaptic weights and do the linear decoding. We know we could do a lot better if we use different types of linear decoders. It's rich information pool. We could encode potentially better if this is the only thing we wanted to know. But it does say, within this population, if you want to get out direction and if you want to get out the speed information, you can do that. Our use case for this is to be able to inform smooth pursuit, but you could basically use the same principle to encode a bunch of information into any network and then try to decode it. So what I told you today, sort of recap, we're working with design principles from systems neuroscience, standard things that we know from physiologist systems neurophysiology. We're using a motion energy model, which is standard out there. It's not something we've invented. We're just implementing it in spikes and dealing with some of the complexity that comes along with that. And then one of the ways that we're using to make sure that we still got a good signal is to do this inhibitory STDP, to do the excitatory inhibitory balancing, and at the end we're using a linear decoder, which is sufficient to be able to extract some of that information back out of a population of cells. And once again, I want to thank my collaborators, Micah and Jerem, who worked a lot on the network and the analysis with me, and Eugene, who is our guiding spirit and CEO at Brain Corporation. Any questions?
You were saying, talking about as if you had real-time adaptive synapses during the excitatory and inhibitory interplay. But the diagrams show GABAergic neurons with excitatory input from the primary cell, and then looping back with inhibition onto the source of the excitation.
Are we talking about this diagram? Is that?
Yeah,
Okay.
Right. That kind of diagram.
Could you say it again? Which one are you referring to? There's kind of two paths of inhibition. One, which is sort of, there's a feed forward volley, which is sort of driving in this direction. This one can get there faster. And then there's this volley, which is basically something like you can drive, this is an inhibitory neuron, and there's some weights which are going to drive this population over here, and then this population response can drive back onto different neurons, but ones of the same class. Yeah.
And that all makes sense to me. So I'm using that kind of as my template for understanding. But then you start talking about how instead of mentioning the GABAergic neurons as being the responding, you were talking about adaptive synapses. In other words, the synapses either of the inhibitory cells, the synapses and the inhibitory synapses themselves, as opposed to let me see if I can phrase this, as opposed to the excitatory inhibitories, let's say to the top GABAergic cell, back down to the AMPA-ergic one. We're talking about the synapse itself as adaptive in some way, so that you could speed things up for the inhibition or something. I got a little...
Okay, so I'll just sort of make sure we're on the same page. So this one right here, this is providing the GABA response. These guys are providing the GABA. Then we have our AMPA drive coming right here. So these are the two different sources that are canceling one out over time. And I did mention something about changes in the speed. In biology, a lot of this might happen really quickly, but in our simulation, we have a sort of step, and we'd like to be able to integrate information. And so what we've done is we've spread, purely in the spike timings, we've spread out the arrival times of these guys in the population. So this one arrow here actually might end up being like, this is like all of the synapses that are coming in from the LGN. So this is a giant population of them. And if you just were going to make a distribution of the arrival times, they have a certain time evolution, the sort of time course. And what we've done is we've spread that out over time. I mean, we actually were using a retinal model that includes some latency coding, and that's going to achieve some of it, but it didn't give us enough for our needs in this case, and so we played this trick of actually delaying some of them with a fixed delay, and that allowed us randomly. We're not, for no particular reason, we sort of, sparse subset is coming later. And so those neurons that come later have the benefit of being activated in a context, and the context is of the other guys having gone earlier. I'm not sure if I'm getting,
Yeah, what you're saying makes good sense. It's I thought you had some mechanism that was, which I was unaware of, for speeding up or slowing down the synapse itself, as opposed to the circuit.
We're not changing the delay times of the synapses. Those are fixed in time. It's just their magnitudes. Okay. Great. Any other questions? All right. Thank you.
three thoughts. The first has to do with our progress in brain science versus our progress in technology. The second has to do with policy, big science versus PI science. The final has to do with international science. So, as many of you know, I grew up in the Caltech of the 1970s. And in my dad's building at Caltech at that time, Christoph was there as a very young assistant professor who was as cool then as he is now. John Allman was there, Mark Kanishi was there, Seymour Benzer was there. It was a pretty exciting time. In my dad's lab at that time, the state of the art was a set of PDP-8 digital DEC computers that were running real-time data acquisition from multiple unit electrodes in freely behaving rats. The computers of that time were, to say the least, incredibly limited compared to what we have now, but at least they were actually able to acquire data using the similar sorts of interfaces that we use nowadays. On the other hand, the conventional wisdom might be that our knowledge of neuroscience, 40 years ago, was at the baby stage. So I remember around the dinner table, the conversation was always neuroscience, and the topic of the hippocampus would constantly be coming up, the context of six-layer cortex, the subject of six-layer cortex, neocortex would come up. Minus from all of that is the 40 years of funded NSF, NIH, DARPA, ONR research that's happened between those times. So one might imagine that the technology would be recognizable and the neuroscience of that era would be unrecognizable. And what strikes me today, especially after a very exciting, wonderful meeting, is that actually the reverse is the case. So if my dad were alive today, so he passed away in 1976, and I were to show him this and tell him the RAM in this, tell him that it was connected to every other computer in the world, he would pop me in the shoulder and go, "Get out of here." On the other hand, if I were to tell him about our current theories of how synaptic transmission is plastic, how synapses communicate bidirectionally actually with one another, that co-neurotransmitters exist, if I were to talk to him about molecular signal transduction, if I were to talk to him algorithmically about what neocortex might be doing, and how neocortex might be, approaches to simulating neocortex might be taking place here in 2013, he would recognize that conversation, and he actually probably productively could time travel to right here, right now, and be a productive member of our symposium that we've been having here. So that's an odd thing. The technology, in fact, is the unrecognizable part, and the neuroscience is the really recognizable part. And in a sense, that's sort of frustrating and depressing because it indicates that, well, as many of you know, the NIH alone spends about $5 to $6 billion annually supporting brain research, and that's to say nothing of other agencies around the world. And yet, our conversation about how the brain works and our questions about the brain, such as how does six-layer cortex really encode cell assemblies and thereby encode concepts and thoughts. How does learning and memory really work? What is the purpose of the hippocampus deep down? You know, a lot of those debates are still here and are still not resolved. So it strikes me that if one good thing can come out of the meeting, this meeting, it's a resolve to actually make as good progress on the neuroscience side as we've made on the technology side over the last 40 years, and to rededicate ourselves to actually making some substantive, powerful progress, so that if I could time travel magically 30 or 40 years in the future, I would be in awe of what had been discovered about how the brain works. It would be a conversation that I could sit and observe with wonder, but it would not be a conversation that I could productively take part in. So that's my word of caution. Now, with regard to public policy and science funding, the reality of public policy and science funding these days is that in our country, the U.S., the Congress and the executive branch have to work together to decide every year to appropriate, that's not authorized, appropriate means actually spending cash, money on the research enterprise that we are all here and we're all about. I've spent 30 plus years in Washington, and I can tell you that what drives neuroscience's special place at the trough is the public health side of that equation, right? So at a deep level, and I say this having worked on Capitol Hill before I went to graduate school, at a deep level, what drives that funding process at the federal level is that members of Congress realize that at some point they or a loved one may get sick with a brain disease, and that they want that special service at the Bethesda Research Hospital that we call the Clinical Center, the NIH. They want the advantages of the public health machine that offers the promise of cures, or at least amelioration of symptoms in brain diseases. And they vote yes. And that's true not only for members of Congress, who have to vote yes for money to be appropriated, but it largely is true for the executive branch and the many, many folks who are dedicated in the executive branch. Their passion and what drives them to support neuroscience year in and year out is the public health side of the equation. Now, in my talk yesterday, or was it the day before? I'm confused. Learning and memory. In my talk, I talked about the economy, and that's the other side of the equation. So Vannevar Bush, no relation to President Bush, was an advisor to Franklin Delano Roosevelt and President Truman. And he actually was one of the parents of the National Lab System that many of you are part of. Vannevar Bush's notion for science investments post-Second World War went thusly. He saw what had happened with the Manhattan Project, and he said, look, if we can continue investing federal money in science like we did during the Second World War, where the raison d'etre is public health, and we spin the technological advantages of that public health research off into the private sector, that can be an economic driver. And by the way, I still resonate to that idea. I find it incredibly powerful, and I believe that it is an important reason why American science has done as well as it has since the 1940s. But the economic side of that equation has become increasingly important. And here's the reason. I presented to you a notion of the great stagnation in my talk the other day. And ultimately what that comes down to is the fact that on the economic side, the U.S. is becoming increasingly stressed. And I would argue to our colleagues and close friends in Europe that on the economic side, those stresses are not just localized to the United States. Those economic stresses are a global phenomenon. that perhaps are a result of the linked complex adaptive systems that we currently find ourselves trying to understand and perhaps not control. But the economic side of the equation is the other half of the Vannevar-Bush promise. The notion that the spin-off from the public health side would drive the economy. And I would submit to you that this workshop that we've been at the last couple of days is about the economic side of the equation. It's different from the public health promise. It's the other half of the Vannevar-Bush proposal. And it strikes me as the area for the potential greatest productivity, lowest hanging fruit. And I would submit that if I walk away from anything, it's the notion that whatever we're working on, as we work on our various projects, the prize to keep our eyes on is the economic stimulus that represents the eventual promise of our collective work. And I would argue that that economic promise from Vannevar-Bush's standpoint extends to the entire population and is not simply represented in wealth disparity, but is actually represented in the happiness of the population and the well-being of the population. Now, my final point, let me talk about international science. I've spent a large part of the last year traveling on behalf of the U.S. government, looking at neuroscience investments globally and thinking about what would be the best U.S. investments over the next 2 decades. So it's a long time scale. So three thoughts come out of that I think are important, particularly since we've had an international flavor to this workshop, and we've had at least a window into what's going on around the world in this field. First of all, happy times are non-correlated. This used to be a real estate stain, by the way, before 2008. But in 2008, happy times actually became correlated in the United States, and we know the results. But at present, still with regard to science, and neuroscience investments specifically, they're non-correlated, which means that around the globe, there are opportunities, and I say this specifically with regard to young people who are in the audience. There are grand opportunities to pursue your dreams, but they are global and they require a global perspective. The second is has to do with specialization. So the neuroscience enterprise across the globe does not have to be a global let's get there first space race, but in fact can be a cooperative effort where we actually collectively work to understand what's going on with the brain, to reverse engineer the brain, and to actually fulfill this public health promise and this economic promise by playing each to our strengths. An example of strength, I spent a lot of time thinking about cognitive neuroscience and functional MRI imaging, which didn't really come up so much at this workshop. But typically here in the States, our studies are statistically underpowered, and that's just the way we run human subject studies. We run them where our human subject guidelines require us to have relatively low ends. And so compared to China, we're statistically underpowered. That gives China a particular advantage in cognitive neuroscience from the standpoint of having thousands and thousands of individual subjects in cognitive neuroscience experiments. But there are many such examples across the globe. This is just one example. But the notion of specialization, I think, is actually very important and can lead to, I think, more productive collaboration where we get out of sort of the space race mentality. I don't think that's entirely productive. I'll end with one other last point about international collaboration. I was a 10-year-old in Moscow during the height of the Cold War in 1966. And it was a pretty tense time to be in Moscow. from a political standpoint, it was kind of frustrating for me because I was into McDonald's and I was definitely into Little League Baseball, and there wasn't a lot of that. But it was very important that there was science collaboration going on at the height of the Cold War, particularly actually in the area of neuroscience between Moscow and the United States. It was difficult. There was an underlying geopolitical game going on, but often at the scientific level, there was genuine scientific interest. And I would say that vignette from the 60s actually repeats itself over and over again today, and that there's an unexpected peace dividend that comes about from global science collaboration. And in that sense, that's an extra benefit of what we're all about. And so I'll leave you with that sort of an optimistic side to all of that. And thanks very much to the organizers for inviting me. And I found this a very, very interesting time. So thank you.